All right,
So, oh,
go
yeah, go
ahead.
ahead.
Okay, I'll go ahead. Yeah, I guess then we'll just go for it with presentation, and then I think the expectation is that the other group, each of the students will be asking a question and sharing one thing that they learned.
Well, I guess in our case, yeah, we only have one student, so
Okay,
he will
cool,
do that.
cool. Um, so I don't know. Who's gonna go first?
We can go first if that's okay.
Sounds good.
So, yeah, so I'll play a video of our missing student, and then we'll transition to Jonathan, who's here today. We and it should be, yeah, we should be able to do that pretty smoothly, hopefully. So I'll share the screen here now. Let's see. All right, so I will. Please let me know if you don't. You should hear audio, but if there's something wrong, please let me know. And yeah, okay, so I will get this started.
Okay, welcome to our SRC presentation. We're studying the metabolic reconstruction of jellyfish-associated microbiomes, or also known as what we did for half the summer. I'm Johnny Nataro, and I worked with my lab partner, Jonathan Ong, under Aki Odera of the Juntoro Lab and Garrett Gibson. So we're studying the Ephyra, the stage before the fully fledged jellyfish, the Medusa. So the Juntoro Lab studies regeneration, and we're interested in understanding regeneration in animals that don't tend to regenerate well. And some of our current experiments use the Aurelia rita and the Ephyra stage, where we amputate some of the arms and then apply different treatments. So we have our control group, and then we have two groups treated with two different antibiotics, vancomycin and penicillin. In the control group, there wasn't a lot of observed regeneration, but in the antibiotic group, we noticed significant changes in regeneration as seen in the graphs and the photos. So we also noticed that two species tend to dominate the Aurelia microbiome. And because we used antibiotics, we investigated the microbiome using the 16S ribosomal DNA to identify the jellyfish and its associated species. We found that the Ephyra stage was actually dominated by two different bacterial species, here highlighted in purple and orange. Under the antibiotic treatment, they exhibited vastly improved regeneration, and the relative abundance of these two species shifted as we noticed more purple bacteria begin to grow in the graph. And comparing the 16S sequence against a known database that we had, we believe the two species are closely related to the Spiroplasma and Ehrlichia, hence the Spiroplasma and Ehrlichia-like designations, respectively, purple and orange. So questions for this summer mainly dealt with modeling the metabolisms of bacteria. We were always working towards a larger goal, and that was to, we also wanted to use a computational approach by doing this. This means that we assembled the genomic sequences of the two species by taking publicly available data and then collected that to sequence jellyfish genome. And again, the goal is to enhance regeneration in non-generative animals, and we also wanted to use something called community flux balance analysis. And what that refers to is kind of the groupings of all the organisms related to the organism, basically combine them to see what they're meant to look like. Had two species that we mentioned before. We have the Ehrlichia-like, which belongs to the order Rickettsialis. It's gram-negative and has a cell wall, and 0.4 to 1.2 micrometers in diameter. It's known to infect insects, dogs in the species Ehrlichia canis, as seen in the picture, and even humans, Ehrlichia murus, which causes a syndrome called ehrlichiosis. Ehrlichia's genome is about 1 genes in spiroplasma. but it's still then we have spiroplasma like spiroplasma molecules. It's gram-positive and is unique in the fact that it has no cell wall. It's about 0.15 to 0.2 micrometers in diameter, and its genome is actually relatively small compared to the rest of its genus. Spiroplasma's genome is about 400 hosts, including insects and plants. They're all endocellular, which means they live inside of the cell. And this encompasses a wide range of parasitic bacteria, and we found this in the jellyfish Aurelia. Okay, so we're going to touch on the metabolic pathways a little bit. And what that means is it's the map of all the pathways in a bacteria's genome. And we have the Ehrlichia and Spiroplasma pathways here. So for Ehrlichia, you'll notice that it has more pathways than Spiroplasma does. But the two pathways are still rather small, and this shows evidence of something called reductive evolution. Reductive evolution is the process of an organism losing many of its genes because it can rely on its host for nutrients. And so because these genomes are rather small and appear to have lost a lot of reactions, we definitely know that they're parasites, and they likely use their host for nutrients and other things that it can get easily, rather than having to synthesize it themselves like other bacteria might. Just for a sense of scale, we have a portion of the bacteria E. coli's cellular pathways. So E. coli, it has many more pathways because it's a more independent organism. And what this means is that it doesn't have to rely on a host to produce its own pathway, so to produce its own nutrients and other types of things it needs to live and grow. But this means it has to synthesize many more compounds. And so as you can see, there are many, many more reactions. And this does not show evidence of reductive evolution because there are more independent organisms.
Okay, so going into flux balance analysis, flux is the movement of metabolites, and flux through a reaction means that metabolites are going through a reaction. Flux balance analysis is a way of simulating the growth of a bacteria. To run flux balance analysis, you need to prepare an FBA file with inputs of biomass, nutrients, and secretions. Nutrients are things that are given to a bacteria, secretions are waste products from the bacteria, and biomass are things that are produced and collected that we use to track its growth. The picture on the left is a simplified representation of a cell with 5 biomasses and 6 reactions. The black dots are biomasses and the white lines are reactions. All of this information can be put into a matrix, as shown on the right, which can then be solved to equal 0 to determine a cell's growth under a steady state. To create a metabolic model, we need to create an FBA file that has flux through as many reactions as possible. When creating an FBA, you often have reactions that are blocked, which means that they have no flux and aren't included in the model. To fix blocked reactions, we look at the login solution files that are generated after each test run. In example A, a metabolite isn't being produced, which means that every reaction afterward can't be used. To fix this issue, we can add metabolite A as a nutrient. In example B, a metabolite isn't consumed, which means that all reactions leading to it won't be used. To fix this, we can add metabolite H as a secretion. Now this might seem easy, but these are easy examples. Most blocking metabolites are harder to trace and can block hundreds of reactions. Using this approach, we were able to generate rough drafts of the models for Spiroplasma and Ehrlichia. With Spiroplasma's simple genome, it lacks genes for glycolysis, TCA, and sugar transport in order to produce ATP. Few transport reactions are presented in the model, suggesting that it uses enzymes to break down recycled peptides and peptides from the host. We were able to incorporate the arginine deaminase pathway, which is shown in the picture, as the main source of ATP production in Spiroplasma. Most of the flux is found in the reactions of the arginine deaminase pathway. We got this to happen once we optimized the model for the importing of ADP and arginine. We had an interesting problem with adenyl kinase, which converts ADP to ATP, being a primary producer of ATP. Adenyl kinase is normally an enzyme involved in homeostasis and not a main source of ATP. We fixed this problem and found that Spiroplasma generates large amounts of flux through ammonia. We also found that almost all necessary nutrients are acquired from the host, which suggests that Spiroplasma is highly parasitic. Now, moving on to Ehrlichia. Unlike spiroplasmas, Ehrlichia's genome has most of the glycolysis and the TCA pathway intact. The top picture is of glycolysis and the bottom picture is of the TCA cycle. The dotted lines indicate reactions that Ehrlichia is missing the enzyme for. For example, Ehrlichia doesn't have an enzyme to break down glucose. Ehrlichia also lacks the enzyme that converts pyruvate to oxaloacetate to power the TCA cycle. So we predicted that glucose was not the main source of ATP. The literature suggests that Ehrlichia should produce ATP from an alternative source, possibly glutamine. We tried to produce models that could produce ATP through other methods but failed. We couldn't create a model in which ATP is produced from glutamine, but we were able to make a model that uses an alternative method. Our alternative method was the import of ribulose as a carbon source. As you can see in the left picture, ribulose is converted to glyceraldehyde 3-phosphate, which powers the rest of glycolysis. We ran the model under different amounts of ribulose, which is shown on the graph. From this, we found that the optimal amount of ribulose for growth is around 1.5 or 1.6. Increasing ribulose doesn't lead to more growth, which suggests that Ehrlichia needs pretty precise conditions to grow in. Taking all that we've learned into account, we formed a hypothesis on how Ehrlichia and spiroplasma affect regeneration, which is shown in this model. The spiroplasma model suggests high amounts of flux through ammonia. Ammonia is known to cause inflammation in cells, and inflammation is known to drive regeneration in some animals. In the earlier graphs, we saw that the amount of spiroplasma increases relative to the amount of Ehrlichia under antibiotic treatment. Since antibiotics increased regeneration, we have a theory that Ehrlichia is somehow inhibiting spiroplasma's production of ammonium under normal conditions. Under antibiotic treatment, we see an increase in ammonium and an increase in regeneration. As for our next steps, we still need to improve our models. We particularly need to understand how Ehrlichia may affect its host. We can then perform community flux balance analysis to model the interaction between spiroplasma and Ehrlichia. Ultimately, we would want to use these models to design experiments to understand how regeneration is affected by bacteria. And I'd like to end this presentation by saying thank you to a few people. I'd like to say thank you to our mentors, Garrett and Aki. I'd like to say thank you to Dr. Leah Goentaro and the Goentaro Lab, and Mitch Eichen and the Summer Research Connections Program. Any questions?
All right, I guess we'll open the floor to the high school astronomy.
Question.
Go ahead, Jonathan.
So on one of the slides, you had a graph with like bars and there's purple and orange like this.
Yes.
And as it went down, there was more of one color. What was like happening as you were going down? Like I saw there was different, you were adding different things to the organism. But like, why was it increasing? Is it like a certain, or was it just the same? Like, are the 3 control and the 3 pesticide? Or I can't remember exactly what.
So the ones at the bottom are the ones treated with antibiotics, and as we treat it with antibiotics, we notice there's more of the purple color, which is Ehrlichia. No, wait, that's spiroplasma. Yeah, that's spiroplasma, and I'm not exactly sure why those increased.
No, but I'm just asking, like, is there any difference between the 3 controls, or are they just the same?
I think they're all the same.
The same, okay.
Yeah, they're just replicates.
Okay.
I have a question. So was there a specific reason you used, I think you said the vancomycin and penicillin versus, I don't know, something else? Or was it just like, you know, oh, I guess we'll use this just because?
I think that's a question for you, Aki.
Yeah, so those are largely chosen because it's what we had on hand. There are other antibiotics that we can now go back and test, but those were chosen prior to knowing what species of bacteria associate with the jellyfish. So future experiments will use other antibiotics.
I had another question. What are pathways? What does that mean?
Oh, pathways are like chains of related reactions that are related to a specific task. So glycolysis is a pathway that's related to the breaking down of sugar into ATP and energy. And
So
the
if
TCA
something
cycle,
has more pathways, it kind of is like more complicated, or it does
yeah,
more.
it
It
has,
has more functions.
yeah.
Okay.
Mhm.
Okay. I see.
If you looked back to if you remember the pictures with all the lines in them, each cluster of lines is a pathway, and each line is an individual reaction.
Okay. Okay.
Any other questions?
Woo! Great job.
Should we all
Am I
unmute for a
done?
round of applause?
All right. Shall we move on to the astronomy talks? All right. I think it's Ian who's going to share the screen, right?
Actually, Isabella was going to start because she has
Okay.
a file that's like just like it's like a personal one that just she can access. So she's going to start sharing with her part of the presentation.
Cool. Isabella, take it away.
Okay. Sorry, this is my first time sharing a screen on this computer, so. Darn. There we go. Okay, can everyone see my screen right now?
Yes.
Okay, so hi, my name is Isabella, and my topic is the selection of stars for optical and radio telescope monitoring with the overall WA. And so essentially, I studied the average spectral types of stars and their stellar properties, such as their flux, luminosity, and their relative flare amplitudes. So the spectral type is the group in which a star is classified according to its spectrum. And the spectrum ranges from hottest to coolest, beginning with O dwarf, which averages at about 30,000 Kelvin, and M being the coolest at 3,000 Kelvin. And so these stellar properties, beginning with flux, is the brightness of a star and can be determined by the amount of energy emitted per unit time per area. And the stellar luminosity is the total amount of energy radiated and can be determined by energy per time. And so the relative flare amplitude is the luminosity of the flare divided by the stellar luminosity. And the stellar luminosity is the constant in this situation, and it's this right here, the total amount of energy radiated, and the luminosity of the flare is energy per time, basically the numerator. And energy being this range of flare energies, beginning with 10 to the power of 31 ergs, and time is the denominator with flare durations from 10 minutes to 100 minutes. So the energy minimum, again, is 10 to the power of 31 ergs, and the energy maximum is 10 to the power of 38 ergs. And the time minimum and maximum is 10 minutes and 100 minutes. So basically, I had four different values per spectral type, which represented the flare amplitudes, and each one represents here the flare luminosity and the flare flux. So if you can see these graphs on the right, the one below it explains here the position, I guess, and each star, let's say right here red, the red star represents the M dwarf, which is the coolest, and the violet represents the O dwarf, which is the hottest. And so these two graphs represent the relative flare amplitudes. And so here I have an Excel sheet, hopefully it does show. Here we go. And this Excel sheet basically shows all of my calculations and how each one comes to equal each one. And so again here, the stellar luminosity, energy per time, the flux, energy per time per area, and the relative flare amplitude. So here are the values of the relative flare amplitudes. And again, here's the constant and the luminosity and all. So yeah, that's my portion. Hope you all understood that. Thanks.
So I guess we can do Q&A for Isabella at this point. Question.
Do we all unmute for a round of applause first? Woo!
No questions?
I have a question.
Okay.
So one of your energy units was ergs. I've never heard of that. Do you have a way of putting it into scale for me in terms of something I might know of?
I'm not sure I could explain that. Let's just say Marin explained it to me in this way. It's just it's an easier way to explain a really big number. So
one erg is massive?
I'm sorry?
So one erg is a massive amount?
Yeah. I wouldn't know exactly to explain it to you, but
yeah. I'm happy with that.
Okay.
Thanks.
Can I ask a question?
Yes.
So how do you collect data on star, or where's this data coming from?
Yeah, so most of this data is from basically their average values. So the spectral type, like the temperatures, they're just average values that I found on different websites. I don't know if you saw the links that I put right there next to the calculations. And yeah, they're all just average values, like the radius also from different websites, and from there I based my calculations off of these average values of the spectral type. It's all based off of the spectral types.
And then, sorry, can I ask one more question?
Mm-hmm.
So this is a way of classifying stars, right?
Yeah.
What do we learn from, or why do we classify stars? What do we learn from that?
To easily place different stars. Basically just to have a better understanding of where each one goes, or I'm not sure. Basically the hotter the star, so again with the beginning with the top of the spectrum with O, is the brighter the star. So I guess
okay.
Yeah.
Thank you.
All right, cool. Ian, go ahead. I think you're up.
Yes,
right?
yes. All right,
Okay, cool.
I'll share my screen here. So first, I just wanted to give, I guess, kind of a little tour of my project. So what I did was I made this website that just shows different data about this star. So here's the name, and here's just some metadata, the coordinates of the star, distance, distance from the Earth, and its temperature. This is a video from the OVRO LWA telescope array, radio telescope array. And then this colorful data plot up here is also from those same telescopes that basically shows the flux and frequency of the radio signals given off by the star. So this is, the color is indicative of the flux, and the frequency is just the radio frequency. And the scatter plot down here shows just flux readings from the star given by the TESS telescope. And these little red dots you can see from this legend up here are actually flares that have happened from the star. And that's basically when the star gives off an emission, it disperses a lot of energy, and the telescope can see that. So that's basically what this graph is showing. So I wanted to talk a little bit about the science behind this. So you can see from this diagram here, this is a solar flare, and it interacts with Earth's magnetic field. Now, our magnetic field acts as a sort of shield from the radiation that is dispersed from these flares. Without it, all that would reach the surface, and that's not good. So, but how that relates to this is that we can see from the flux of the radio data and this scatter plot, if there's a large, if there's a flare along with a high flux in the radio, it's likely that that's a CME, which is, it stands for coronal mass ejection, and it's basically a very large flare that is put out from this star, and the importance of that is that we can see when a flare interacts with a planet's magnetic sphere. As I had said before, our magnetic sphere protects us from the radiation of these flares. So if we can see it on another planet, then that's a sign that planet could possibly be habitable. Obviously, it's not a definitive thing, but it's another variable to the equation to say, hey, you know, this planet could possibly harbor life.
Cool. Questions? All right. Let's make for a round of applause. Thanks, Garrett.
Yeah.
Now questions.
That was a really nice website interface there. Are those two data sets actually overlapping at all, or are they just examples, I guess, for now?
Well, this the image is just an example, but this was actual data. Because this is the here I can actually share again, so I can do use my pointer. So this one up here is just like a still image But this is actual, this actual graph. And I'm realizing now I forgot to show that this was probably the most involved thing when I was programming the website. This actually uses a library. And one of the features is that you can like select a portion of the graph and it'll zoom in on that portion. So you can look around and look at, I guess, more exact like portions of data. And yeah, so this is individual data points that this is pulling from.
That's awesome.
I have a question. So those flares in the figure, are they manually declared flare as a flare, or is that, do you have a threshold that
Well, I have a in the code, I have an array. There are three arrays. One is the flux value, so that's on the y-axis, and then there's the time, and then the third variable is just true false booleans. If it's true, then that means it's a flare and it's marked as red. If it's false, then it's marked as blue, because that's just a flux reading, and there's not a flare.
Okay, so yeah, so I was wondering, there's that sort of a few dots next to the far right red flare. And I was wondering why that one isn't considered a flare.
Sorry, what do you mean?
Yeah, so if you go a little bit more to the right with your cursor, why that's not, that column isn't a flare?
Well, that's just like energy given off, because there, it can give off different levels of energy, or different levels of flux, which but it doesn't necessarily correlate with a flare from the star.
Mm-hmm. And then, sorry, the video you showed to the left on the web page, what is that exactly? That's a star?
It's basically it's radio. It's like radio frequency. That's just visualized. So this is from the radio telescope array.
I see. So we're looking at space right now, and those dots in there are the stars. Okay, just making sure that we didn't have the technology to get that kind of image of a star. Okay.
No, it's not. It's just the radio frequencies.
Okay, awesome. Yeah, thank you.
All right. Who's up next? Is it Jaden?
I think so.
Okay, yeah. All right, go ahead, Jaden.
So I was tasked with doing a research summary. So we were tasked with familiarizing ourselves with the basics of observational radio astronomy and the classification of transit candidates based on their spectral readings and images. So I'm going to explain all of the research I've made into a short lesson. The Owens Valley Long Wavelength Array is designed to study high redshift as a probe of the epoch reionization and fast cadence imaging of the sky for the low frequency transients. This is aimed at the detection of coherent radio emissions from extrasolar planets. For certain terms like high redshift or epoch of reionization, below gives a short definition. So coherent radio emissions from exoplanets can be used as an example, and compact object merger events such as stars combining. Throughout this time, it focuses on solar dynamic imaging spectroscopy, which is the measurement of spectra produced when matter interacts with or emits electromagnetic radiation. This Wavelength array opens up the field of radio transits, performing targeted transit searches and blind surveys to better sample the transit phase space identification of new populations of radio transits, detection of extrasolar planets through their auroral radio emissions, provides measurements of magnetic field strength and rotation rates, and directly serving as a probe of interior composition and dynamics. That's all.
All right, cool. All right, round of applause for Jaden. And questions?
Can you tell me more about the transient phase? What does that mean?
Sure. I had to cut down a lot of my research notes into a short summary to fit in the slideshow, but I can pull up my notes as my explanation.
Yeah, that'd be fine. Either pulling your notes up or just saying what you know from memory.
Yes, I will do that. I'm sorry, I have to type from my phone and to reach my notes. It's a little slow. So transit searches kind of act as a survey to review data from events taken outside of space using different radio telescopes to find extrasolar planets.
Thank you.
I have a question. I don't know if you know this, but how often do we find exoplanets using this method? Or I guess when we say we, astronomers in general.
I'm not sure if I have the statistics on that and my notes, but I can check.
Sure.
I can interject on this one. We've never found an exoplanet through their radio mission. We know that the solar system planets emit in radio waves, but it's a very hot topic right now, trying to use this method to detect exoplanets.
Oh, okay.
We do have a 13 Jupiter mass thing, free floating planet that has been found as radio bright. Not discovered via those means, but was detected as a radio source similar to the planets.
Oh, wow. Okay. Awesome. Thank you.
Okay, cool. Thank you, Jaden. All right, let's move on to Jonathan. Astronomy, Jonathan.
Yeah.
No.
That is that. Yeah, let me share my screen.
Also, hi, Mitch.
Okay. You guys can see it? Yeah.
Yep.
Okay. Yeah, so yeah, I'm Jonathan. My research mentor was Professor Hallinan. And my project was about determining the optimal placement for the Farside radio telescope. So what is Farside? So Farside is this radio telescope array it's currently being proposed to NASA to be to install a radio array on the far side of the moon, because the moon is tidally locked to the Earth, so we only see one side of it all the time because it rotates and revolves at the same rate. So there's a far side that never faces the Earth. So some of the goals of FarSight are to detect CMEs, which Ian was talking about, these coronal mass ejections from other stars, and detect planetary aurora, which is evidence of a magnetosphere. So just like how on Earth, when CMEs come and hit against our magnetosphere, we get auroras at the northern and south poles. And so this actually glows in very low radio frequencies that we want to be able to detect, because for example, Mars, we believe it used to have a much thicker atmosphere, but these CMEs, what they are these huge blasts of plasma from the star that just get blasted out into space, and they slam into the planets, and what happens is the atmosphere just gets blown away from them unless they have this magnetosphere protecting them. So Mars used to have an atmosphere, but now it lost a lot of that because it doesn't have that magnetosphere. So the reason that it's kind of a hot topic right now is because it's kind of, like Ian said, is redefining like habitability. You know, it's not just about whether we can make water. It's, you know, to have an atmosphere, we think we need that magnetosphere. So we're trying to see, you know, if other planets out there, you know, what, like, how are their magnetospheres? Do they have them? Stuff like that. So the reason we want to put it on the backside of the moon is because in radio, to be able to detect these magnetospheres, we have to get away from the Earth auroras, because, you know, if we're trying to detect one of an exoplanet, like, you know, billions of miles away, and there's one, you know, just a couple 100 miles north, right? It's going to totally wipe that out and we're not going to be able to see it. So we need some kind of radio protection in a way. So as you can see in this diagram, you know, right here on the backside of the moon, that's where the radio frequencies are the lowest from the Earth, you know. And plus there's also commercial radio, right? So our FM and AM radio are causing interference. So the best place to keep it, to look for these very low detections, are on the backside of the moon. And this is just kind of how it's going to work. The rover is going to land right here, the module, and the rover is going to drive along these lines, laying down basically radio sensors, and then it makes a big array, and that's going to be the whole telescope. So it's going to be, you know, pretty big thing, 10 kilometers. So my job was to figure out where to keep it on the moon. So there's these, to map out stars in the skies, there's celestial coordinates. They consist of right ascension and declination. So right ascension is like kind of east to west. So like if you know, the Earth is rotating, so the stars are passing by us like that, that's right ascension. And declination is, you know, how close to the poles are the stars. So it's up and down kind of. So what's important to realize is that the moon is orbiting around Earth. So the right ascension actually doesn't matter for the stars, because we're going to be able to see them eventually somewhere in the orbit, since it's more orbiting like that. But what matters is if we're too close to the poles, we won't be able to see in the southern hemisphere, or, you know, vice versa. So latitude is what actually that matters for choosing the best target. So to figure out the latitude, there are 3 steps that I took. First, find data on the planets, on the exoplanets, to choose targets of interest. So I just kind of made that up, targets of interest. Basically it means, you know, how likely are we able to detect a radio signal from them, or an aurora from those planets. This can depend on distance. Mainly it's depend on distance, but also, you know, how brightly it can shine in the radio, you know, and I'll get into that. And then the second thing is to figure out how high up in the sky each of the planets are going to be. So if you're standing on the moon, the reason this matters is because far side is going to be flat on the moon. So planets that are directly above 90 degrees up from far side will have the maximum sensitivity. But if it's very low near the horizon and it's coming in at an angle, you won't get as much sensitivity for those planets, or you won't be able to detect as much. So we want to prioritize the best planets and get them as high up in the sky as possible. So I'm trying to figure out where on the moon I can do that. And so basically determine which latitude gives the highest total flux. So 2 terms you should probably know is that flux is, our luminosity is the energy from an object. So just raw energy coming out of like a star's luminosity. And that's huge, right? But a flux is how much of the energy reaches us. So that depends on distance, basically. And since these things are really far away, the flux is, can be smaller and less. So it's dependent on both, right? So we're looking at the flux, the highest, which latitude gives the total flux in radio. So there are two data sets. The first data set I had was known planets. So these are planets that we have confirmed and we know they're out there. And the which planets I chose came from this paper called Can We Detect Exoplanet Aurora Orbiting M-Dwarfs? So what this paper did was it took different estimates of how strong the CME is going to be and how strong the magnetic field is going to be to figure out, when they hit each other, how brightly will they shine in radio. So it depends on both of these things, right? So they took, different various estimates and combined them, figured out which one, how they're going to shine. They did the calculations. So this is the luminosities of them in radio. So then I looked up those same planets in this archive called the NASA Exoplanet Archive, which is just a big list of every confirmed planet. So from that archive, I've got a bunch of information. So the name, the distance, how far it's orbiting from the star, which is the semi-major axis, and then the coordinates in the sky. So you see celestial, and then there's the ecliptic coordinates. So celestial coordinates mean the coordinates of where they're going to be on Earth, and the ecliptic coordinates mean where they're going to be if you're standing on the ecliptic. So the ecliptic is the plane at which the solar system orbits. So all the planets all orbit in a disk on the same plane. That's called the ecliptic. So since we know the Earth is tilted compared to the ecliptic, so celestial and ecliptic coordinates are kind of different. But the thing is, this matters because the moon and the Earth are tilted compared to each other. So if I'm standing at the North Pole on the Earth and I look up, I'll see Polaris. But if I'm standing on the North Pole of the moon, Polaris won't be directly ahead. It'll be slightly off somewhere else. And converting between those two are kind of a pain. So it turns out that the moon is actually very close to the ecliptic coordinates. It's almost perpendicular. So this is just a pretty good approximation for the lunar coordinates. Ecliptic is just about the same. And then, I got some information about the stars. And so finally what I did was I took the radioluminosities that I got from this paper, how brightly they're going to shine, just how much energy they're putting out. And I figured out how far they're going to be from us and how high up they're going to be at for different latitudes. So for example, if I'm standing at 90 degrees, if I'm standing at the North Pole and I look for Proxima Centauri, how brightly is it going to shine in radio? How far is it from us? And how high up in the sky to figure out some flux that the detector will be able to see from Proxima. And then if I go to 75, how does the flux change? And then to 60, then 45, and so on, all the way down to -90. And I did this for every planet that I had there. And then I just took all the planets and all their fluxes at the different latitudes, and I summed them up at each latitude. So at 90 degrees, this is the total latitude you get at the bottom, and then, at same thing, 75, 60, all the way down, 90 to -90. I summed up all of them. So that's the first data set. Second data set is unknown planets. So these are planets that we aren't sure that they could be there or they could not be. So this is basically just all the stars, so we don't know if there's a planet there. There may or may not be. This is just out to the sample that I chose for the stars, which stars I chose, were just all the stars out to some distance. So this 10.5 parsecs, that's about 30 light years. And so the data I collected about these stars came from the Simbad archives. And this was just the names, the distance, and the celestial and ecliptic coordinates. And so for this one, I did very similar thing. I figured out how high up they're going to be. And depending on how high they're going to be in the sky, how much far side can detect from those fluxes. And I just summed up at the bottom the percentages, So if it's at 45, maybe it'll get 40% of the total flux, whereas if it was 90. And then I did that for all the planets, all the latitudes, summed it up at the bottom. And so finally the results, so it turns out for the unknown planets, the lunar latitude summing to the highest received fluxes is basically around 0 degrees on the latitude. So that's basically the equator, which makes sense. This is kind of expected because if you randomly distribute all the stars across the sky, and you think about, if you're standing on the equator, you can see 90 degrees up and you can see 90 degrees down, and then you're orbiting, so you'll see 360 all the way around. So you'll basically be able to see every star. So that's kind of the just to see every star. But what turns out for known planets, for the planets that we know, it turns out that there's a bias towards the southern hemisphere. So the best place is actually negative 15 degrees latitude. And this is different because we're also taking into account, how brightly they shine and the distance from us. So if there are better targets in the southern hemisphere, that will pull the best latitude a little bit lower, rather than the northern hemisphere. And so finally, if you kind of didn't get any of that, this is the main idea. So green is the best place to receive radio signals from these planets and from the stars, and the red is bad. And this, you can see latitude and longitude here. So on the back, it's just the surface of the moon, lied flat. And these dashed and dotted solid lines are actually the areas that the radio interference is very low from the Earth. So here, inside this circle, that's where you'll get the least amount of, enough like radio silence to be able to detect these objects. And then the green areas are where we want to stand to see the most, the best, planets to look at. And yeah, so basically the answer is negative 15 degrees. Somewhere in this region is where we should put far side. So any questions?
All right, round of applause.
Woo! I'll just keep this diagram up if you want to look at it more, but yeah, any questions?
So this is, oh, sorry.
Go ahead.
Is that 3 at the same time? I think that's a new record. I've only ever had 2. For the unknown planets, you said you set a limit of 10.5 parsecs. What made you choose that limit?
Well, it just kind of arbitrary, just kind of shows, the original was just 10. So then I was like, okay, maybe 10.5, so we don't miss anything. But it doesn't matter too much since it's mostly spread out pretty randomly. And it's around there, around 10 to 15 parsecs that they become too far and too dim for us to detect the aurora. So that's kind of a goodish cutoff, but yeah. And I forgot to mention this graph is based on the known planets, not the unknown planets. Since the unknown planets kind of summed up to be around where we expected, not really too much new information, this is the main deal is like the known planets is an 8, but yeah. Any other questions?
I have a question.
Yeah.
Does the terrain of the surface of the moon matter for these detectors?
Yeah, it will matter. Because if you put it on the side of like a crater, it's gonna fall, or you want it to be a good area. And also, the craters might be too big for this to happen, but I kind of thought if you put it inside a crater, it's like when there's mountains on Earth and like they're covering kind of parts of the sky. But I don't think that'll matter too much. But that's actually kind of the next step, like where in this area exactly we want to land it, and with the terrain and stuff like that. That's the next kind of consideration.
Awesome, thank you.
Yeah.
All right, any other questions for Jonathan?
I have a question.
Yeah.
The process of summing up all of those planets seemed quite tedious. Is there any software that can help you, or did you think of somehow partially automating that process?
Excel was really helpful, because like it was like crazy amount, like 200 planets, 400 like stars and stuff. So, but that little dragging thing just like So satisfying. You just put the equation in the first one and you drag it all the way down and it sums up all of them for you. And it's really nice. And actually right here, I have the Excel's, the links to both of them. I don't know, do you guys want to see that or? I think it's okay, but yeah. So that's, yeah, it was Google Sheets technically, but yeah.
Yeah. Well, thanks.
No problem.
All right, I think that concludes our presentation session. Good job, everyone.
Yeah. Thanks, everybody.
Yay. Yep.
Yeah, thanks. Thanks for moderating, Yuping.
Yep. All right. Also, Mitch is here. Hi, Mitch. It's recording. We're done.
Thanks to Mitch too for the program. It's a really great program.
Yes. Our pleasure.